Hello, good day. I am Angieliquie Mejilla from BSN 1A. Now, let's move on to Microbiology and Parasitology, Chapter 8, Controlling Microbial Growth in Vitro. So, what is in vitro? In vitro meaning outside an organism. So, this topic is about controlling microbial growth outside an organism. So, let's talk first about the factors that affect the growth of microorganisms. What are the factors that affect the growth of microorganisms? First is the availability of nutrients. So, an organism needs nutrients in order to survive. Second is moisture. So, on Earth, water is essential. So, an organism, in order to survive, it needs water. So, it needs moisture. Third is temperature. So, an organism, in order to survive, it needs good temperature or normal temperature for an organism to survive, depending on iya kaya. So, the fourth one is the pH. So, pH refers to acidity and alkalinity. Later on, I will talk about types of organism na makakasurvive. Another is the osmotic pressure. Osmotic pressure is the pressure that is exerted on the cell membrane by solution inside and outside the cell. Another is the barometric pressure. So, some organisms can survive in a barometric pressure. For example, of barometric pressure is an organism that is unable to survive in a deep ocean with the high barometric pressure. So, let's move on to the types of microorganisms. There are types of microorganisms. The first one is the psychophiles. Psychophiles means an organism that can survive in a cold temperature. Another is mesophiles. Mesophiles is an organism that can survive in a moderate temperature. Another is thermophiles. Thermophiles is an organism that can survive in a hot temperature. Next is halophiles. Halophiles is an organism that is able to survive in a salty environment. Haloguric is an organism that survive in a salty environment. Amot tawagan, organism na nakaka-survival, salty environment. Haloguric. Alkaliphiles is an organism that is able to survive in an alkaline environment. Acidophiles is an organism that is able to survive in an acidic environment. Barophiles is an organism that is able to survive in high barometric pressure. Like in mga organism na nakaka-survive ngadtuan deep ocean. So, there are 3 in vitro sites where microbial growth is encouraged. First is in laboratories, microbial growth laboratories. And another is in research laboratories. And another mga industries. Some industries like antibiotic industries or manufacturing of antibiotics, nakaka-survive nga itong mga organism. So, I have here what is the meaning of controlling microbial growth in vitro. So what is culture media, inoculation of culture media, and incubation? So culture media means culturing a bacteria. Inoculation of culture media means adding a portion of a specimen into a bacteria. Incubation means it is a bacteria or a culture media is placed in an incubator. So there are types of incubator that is used in medical laboratories. So types of incubator is the carbon dioxide incubator. Carbon dioxide incubator is an incubator in which a cylinder of an carbon dioxide is attached. Non-carbon dioxide is an incubator in which containing a room air. Anaerobic is an atmosphere. Anaerobic incubator is an atmosphere devoid with oxygen. Anaerobic means without oxygen. So that's the types of incubator. So let's move on to, there are categories of media. So the 3 types of or 3 categories of media are enriched media, selective media, and differential media. So what is enriched media? Enriched media are necessary to grow pathogens, especially fastidious organisms that needs enriched media. For example, it needs nutrients. It's called nutrient agar. For example, a bacteria, kung culture media and bacteria, kailangan din mag-add in nutrient agar. The second is the selective media. The selective bacteria are used to grow specific bacteria while inhibiting the growth of unwanted bacteria. So selective la, i-inhibit an unwanted bacteria nga mag-grow. Pero i-grow it specific bacteria while inhibiting the unwanted bacteria. The third is the differential media. Differential bacteria are used to differentiate the bacteria. So, mean differential. Iyagin, di-differentiate. So, let's talk about why is it important to practice sterile technique in a laboratory? Because if we are going to grow a bacteria, it should be in a sterile environment. We should use sterile technique in order to contaminate the medium. In order not to contaminate the medium. So, we need to practice sterile technique. For example, kung mag-culture media po kita, kailangan mag-practice kita ng sterile technique kay it bacteria na ato nagpapagrow, dapat diri mahaluan hiniba na microorganism like it mga airborne microorganism. Para ito ato nag-culture nga bacteria, hiya gudla, waray na iba nga mag-grow ito nga bacteria, nga mga unwanted bacteria. So, let's move on to bacterial population growth curve. Bacterial population growth curve. There are 4 phases of bacterial population growth. And here is it. The first one is the log phase. Second is logarithmic phase. Third is stationary phase. Fourth is the death phase. So, the first phase is the log phase. This is the first phase. So, in a log phase, bacteria absorbs nutrients. Pero, diri pa po iya mag-grow. Diri pa na-grow it bacteria halag phase. The second phase is the logarithmic phase. This phase, logarithmic phase. In a logarithmic phase, the bacteria, nag-grow na ang bacteria. So, logarithmic, nag-grow na po ang bacteria. Or, rapid growth na. So, in logarithmic phase, it is a rapid growth of a bacteria. Pagka di po stationary phase, ang pag-grow puliwatan bacteria is na slow. Nga di, steady pa. Nga di heavy is na-grow na ang bacteria. And then, after pag-slowan pa ang growth and bacteria, makadinagyap death phase. The bacteria then dies. So, this is the bacterial population growth curve. So, let's move on to inhibiting growth of microorganism in vitro. So, inhibiting growth of microorganism in vitro, meaning the growth of a microorganism is inhibited. For example, mga hospitals, nursing homes, or other healthcare facilities. Kailangan i-inhibit ang growth ng bacteria in order not to infect patients. For example, na hospital. Kung i-inhibit nga ni meaning gimpapastop or stop ang growth han bacteria. So, in a hospital, it needs to inhibit the growth of microorganism in order for the patient not to be infected. So, kailangan i-inhibit ang growth han bacteria. So, that's the end of my discussion about controlling microbial growth in vitro. Thank you for watching.